Hi, this is the final project for microbiology class. This is a micro project, and my name is Dailenis Garcia, and today is December 7, 2023. What I did for this project was looking for information about the three major microbes: eukaryotic, prokaryotic, and viruses through bibliography in biology books, Valencia College database in for the CDC website, Centers Communicable Disease in the United States, looking for the outbreaks, information, and all the six that I'm going to explain by. I'm going to resume all those microbes. I did a big explanation into the slides presentation PowerPoint. And here is little B about what I did in my PowerPoint. For eukaryotic microbe, I choose the Entamoeba histolytic. This is a single-cell organism, specifically eukaryote, meaning their cell contain a nucleus. They are parasitic and can inhibit a wide range of hosts, including all classes of vertebrates, some invertebrates, and potentially other unicellular eukaryotes. Their life cycle is generally simple and consists of two main stages, the cyst stage and the trophocyte stage. The cyst stage is the ineffective stage where the organism is domined and protected by two other cells, allowing it to survive outside the host. The trophocyte stage is the active multiplying state that occurs inside the host. Transmission of Entamoeba histolytic typically happens through the ingestion of this cyst, which can be present in contaminated food or water. Once ingested, the cyst transforms into trophocytes in the host gut, multiplying and can cause the disease. Humans can host at least 6 different species of Entamoeba, indicating the diversity within this genus. Each species might have slightly different characteristics of pathogenicity. The primary treatment for this disease is antimicrobial therapy, typically with medications such as metronidazole, tinidazole, followed by paromomycin or iodoquinol, to eliminate any remaining parasite in the intestine. As for vaccination, currently there is no vaccine for amoebasis caused by Entamoeba histolytic, but research is ongoing, but there is no vaccine approved for this human yet. Therefore, we cannot discuss the side effect of the vaccine since there is no vaccine available. My next microbe is Moraxella bovis. Moraxella bovis is a gram-negative aerobic oxidase positive non-mothing bacterium. It is the primary cause for agent infections in bovine keratoconjunctivitis, also known as a pink A. Bovine keratoconjunctivitis primarily affects the eye of a cattle. However, it's not just the eye that is involved in this disease process. There are other systems also involved, like the immune system, respiratory, skin, and nervous system. Moraxella bovis resides in the eyes, nasal cavities, and other reproductive tract of the cattle. Under normal conditions, the bacteria do not cause the disease and live as a part of the normal flora. When conditions change, such as increase of UV light exposure, dust, or the presence of flies, the bacteria can cause infection. The disease is spread through flies. Flies carry the bacteria from the eyes and an infected animal to AIDS of a healthy one. Treatment are potential. That's why this is a communicable disease, because that can spread through one healthy animal to another, one infected animal to another healthy one. So the treatment typically involves the use of antibiotics to control the bacterial infection, along with supportive care to manage the symptoms and prevent further damage to the eye. The primary vaccine available to protect against this disease is the Moraxella bovis bacterium vaccine. The Moraxella bovis bacterium vaccine is a killed or inactivated vaccine, meaning It contains bacteria that have been killed or inactivated but still stimulate the animal immune system to produce protective immune. From this microbe, there is no outbreak or other major outbreaks since there is endemic disease which affect only the cat population, and since the controls are doing a good job, the spread can be controlled in in small areas. The next one is the yellow fever virus. Is that the family from Flavivirus and have a single standard positive sense RNA genome. This means the RNA can be directly transmitted into proteins by the host cell machinery. These proteins are involved in the replication of the viral genome and evasion of the host immune response. The viral envelope is derived from the host cell membrane during virus binding. It contains the E and M proteins, which are embedded in the lipid bilayer. Primary affects the liver. The liver is the organ where the virus multiplies and causes damage, leading to the characteristic of jaundice, yellow of the skin and the eyes, seen in several cases. High fever return and several body systems are affected, usually the liver and the kidneys. Yellow fever is a viral disease that is spread primarily through the bite of an infected mosquito. The disease cycle typically involves transmission between mosquitoes and non-human primates, like the monkeys, in a jungle or forest environment. Yellow fever virus is a member of the Flavivirus genus. Uses several virulence factors to evade the host immunity and cause tissue cellular damage. These virulence factors allow the yellow fever virus to effectively infect host cells, evade the host immune response, and cause tissue damage, leading to the symptoms of yellow fever. The yellow fever vaccine is a type of immunization that contains weakened form of the live virus that causes yellow fever. This vaccine is used to prevent yellow fever, an acute viral disease, by introducing a weakened form of the virus into the body. The vaccine stimulates the immune system to develop the antibodies against the virus. The World Health Organization estimates that there are 200,000 cases of yellow fever annually. And within the 90% occurring in Africa. The prevalence of the yellow fever is endemic in 34 countries in Africa and 13 in Central and South America. Despite vaccines efforts, the prevalence remains high in these areas due to factors like climate, the presence of mosquitoes, and the proximity of mosquitoes beating the bites and to human habitat. The mortality rate for these vaccines is the mortality rate for these of those who contract the disease, the case fatality rate can be like high as a 50%, who estimate that there are 30,000 deaths annually due to the yellow fever. The major outbreak, which was in 18th and 19th centuries, and several outbreak occurs in North America, Europe, brought by ships from Africa. In 1980s, resurgence in Africa due to the decline of immunity among the population, leading to an epidemic in the West Africa with an over of 10,000 cases. In 2016, large outbreak in Angola and Democratic Republic of Congo within 965 confirmed cases and thousands of more suspected. In 2017 and 2018, Brazil experienced its largest outbreak in decades, with more than 2,000 human cases over the 700 deaths. Efforts to control yellow fever focus on vaccination, mosquito control, and public education to reduce the risk of mosquito bite. Despite this effort, yellow fever remains significantly public health issue in many parts of the world. In conclusion, this disease can be caused by different types of pathogens and affect different organisms. They are preventively to some extent through vaccination, like yellow fever, good sanitization practices like the amoebasis, and management practice to reduce the spread of the disease like the bovine keratoconjunctivitis. This is the brief explanation of what I did in my PowerPoint slides. There is more deep into the details for each part of the question in the PowerPoint, and I hope with this explanation you have a brief idea of what I'm trying to explain and what I did into my project. Thank you so much.